different fungi and bacteria that are present in ASD. For decades, farmers have utilized methyl bromide fumigation as a method for pre-plant control of soil-borne phytopathogens. Methyl bromide fumigation, however, is not a very sustainable method of control, as it is an ozone depleter and is a carcinogen, so it's bad for us and bad for our atmosphere. And for this reason, the EPA discontinued use of methyl bromide in almost all crops. almond hulls, almond shells, spent barley and yeast from the beer brewing process, ethanol, molasses and red grape pomace, tomato pomace, molasses, red grape pomace, tomato pomace, mustard seed meal, rice bran, trub, which is spent yeast, walnut hulls, and walnut shells. Experiments were performed twice for each carbon source and set up in a completely randomized design with 4 replications for each carbon source. Experiments were repeated twice. Each bioreactor has an ORP sensor, which senses, ORP stands for oxygen reduction
Morning everyone, my name is Alex Kwong and I've been spending 6 weeks here in Dr. Claire Castillo's lab in the plant pathology department under my mentor Andrea Chung. And I've been studying the interactions between Candidatus liberibacter, which is a really cool bacteria, and potato virus Y, one of the most common viruses in America, in russet Burbank potatoes. So, just a few hundred years ago, 1790 in America, 90% of Americans were farmers, and now it's only 2%. The reason why we have this, we can do other things besides farming is because we have a bunch of efficient techniques and we have scientists constantly working with the farmers to create pesticides and create things that target the pests in the fields and target these pathogens. So the main goal is to improve crop yield cost effectively, and that's the main point of my study. Usually we study plants and the insects and the disease they bring in a one-to-one relationship. But the cool thing is, in the fields, there are hundreds of bugs at the same time on the plant and hundreds of pathogens interacting. And they cause interactions that you can't really see if you only study one. So my study is studying a bacteria and a virus and how they interact within the plant. So some background information before we go on. This is pretty important to understand the methods later. First, the bacteria, LSO, Candidatus liberibacter. It's transferred by psyllids and it's a bacteria. The way it's transferred basically is the psyllid decides to go feed on the plant, it gets in the plant's gut, it gets around the insect, and then when it goes to a new plant, it can transmit it. The reason why this one is so important is because of two major diseases. It's closely related to Huanglongbing, which is a huge citrus virus in Florida and in Asia, causing millions of dollars each year. So by studying this, we can understand that. Also, it causes zebra chip disease, which is a disease on tubers which makes them unmarketable. So we're trying to learn more about that relationship as well. Another interesting thing is it can impact its vector, the psyllid. So when the LSO gets into the psyllid, it causes the psyllid to have increased fecundity, which is more reproduction basically, and it can affect which plants the psyllid goes to feed. Next we have PVY, so that's the other pathogen we have. It's a virus. It's transmitted via aphids, same thing, it gets in the body and then it allows the aphid to transmit it to new plants. This is very widespread. This is a very widespread virus across all of America. Every single potato plant is tested for it before it can be grown in the seeds. Yeah, so that's all we need to know about that. So this is pretty interesting. The reason why we decided to see it is you can see on the map the highlighted region is where LSO generally occurs. And PVY, like I said, occurs all across America. So this interaction that I'm talking about is happening in this region. And luckily enough, we're in California, so we can study it right here in our fields. If you look at this data from June to July and then August to September, you first notice aphids, the PVY vector, and so there's PVY on the plants, we detected that. And then 2 months later, in August and September, we see psyllids and then LSO. So we think there's some kind of relationship between these because one appears right after another. So the study was putting them on the same plant pretty much and detecting whether the PVY or LSO were interacting and affecting each other. Cool. So simply, the methods get pretty complicated, so I'm going to give you this slide first. First, we inoculated a set of healthy plants, and then we put healthy insects, healthy aphids, and healthy psyllids onto these infected plants to pick up the viruses and the bacteria. And then we moved these infected insects to a new set of healthy plants to see how they transmitted it to the new plants. So by testing to see the new plants that we infected, how infected they were, we were able to see compare and see if the PVY LSO caused a different level of interaction. So in more detail now, this is the first step, inoculation. So we had russet Burbank potatoes, 1 month old. They were originally all 4 healthy. Then we infected one set with PVYO, so it's a PVY virus, and we used a mechanical inoculation, which is basically taking some powder and some of the virus extract and rubbing it on. Also, the bacteria can only be infected with psyllids, so we got infected psyllids, 3 of them, put them on the plant for 72 hours. And the 4th type of plant, we did both the PVY and the LSO method. And we did multiple copies of each of these plants, so there's pretty much, there's a ton of each one. And this study was done in correspondence with another lab in Washington. So they did the first half of these steps, and we did the second half. So moving on, this was also done in Washington, the acquisition step. They put 10 healthy aphids, 10 healthy psyllids on each of these types of plants. So you guys see the bottom row, those are the plants that we just infected. We took these healthy insects and put them on, and hoped that they would receive the virus or the bacteria. Cool, and then we took these respective insects that were on each plant. So technically, some of them should have LSO. The psyllids should have LSO. Aphids should have PVY on them. They should only be picking up the ones that they can transmit. So you put them on the plants, and then you put them on a healthy set of plants, and they should be transferring the virus or bacteria they're carrying to the new plants. the cool thing about the study, going back, is you see the LSO and PVY plants. They should technically pick up the LSO or the PVY because both of them are on here. But when they're putting it on the new plant, we can measure the amount of virus or bacteria on them, and they should have a different amount than just the PVY or just the LSO plant. So that's what we're looking for. So then that was all done in Washington. So this is what we did in the lab. DNA and RNA extractions. So LSO, you can get the DNA for. So we did DNA extraction using the CTAB DNA method. Some people described DNA methods yesterday. It's just pretty much a 3-4 hour process that you just have to repeat over and over. Then we nanodropped it, which basically means you get what the concentration of the DNA in your extract, and then you put water in it to make it all the same concentration, so you can compare it like that way. And then you do qPCR, quantitative PCR, that's what Kane's presentation was on. And you can compare to see within these samples how much of the bacterial DNA we have. So we did the same thing for RNA. PVY is an RNA virus, so we had to use the RNA kit instead. We bought a kit, same thing, took a couple hours. Then we had to form cDNA, which is unique to the RNA, and then we did qPCR to compare. So we designed this experiment from scratch, so you need to constantly fine-tune your methods. Interestingly, set A, the first set of healthy plants which we had to infect with PVY and LSO, only 25% of the plants actually got infected. So in the first place, we didn't have all infection for A, so we couldn't make any definite conclusions for B and C. But we still did get some infection in B and C, and that's what's really interesting. I'll show you next. So the results, set B, PVY. So remember, this is when we took PVY's aphids. We took the aphids that were infected from their plants, and we put them on another healthy set of plants to see how well they would transmit PVY. Control doesn't have anything, as you would expect. Also in PVY's, or we'll start with PVY. PVY transmitted. There's high error, but we did get transmission, which is what we expected. LSO doesn't have anything, which is should be as such because they don't transmit LSO. LSO plus PVY also is very low, and we don't know whether this is due to the fact that we didn't have infection on A, or if it's because LSO and PVY were interacting in such a way to suppress the development of the virus. And then the results for set C, this is when we took the LSO vectors and we put them on a healthy set of plants. So technically we should only be seeing LSO infection here. Control, that's our control level. LSO was infected, which is good. For some reason we saw infection for LSO on the PVY plant, which is really weird. So either there was contamination or it's possible that the vector transmitted LSO for some reason, the PVY vector. So that would be really cool. That was recently discovered in the labs, that a different organism was transmitting something we didn't expect. So that could also be what's happening. And then LSO PVY again, we don't know whether it's because there's a lack of infection in set A or because something weird happened and some interaction occurred. So a few things to improve this for next time. First of all, we had 10 samples for each type of plant. So we need to finish all 10 samples pretty much. Right now we've only done 5 out of the 10. But based on our results, I don't think anything different is going to happen. Set A, like I said, needs to be finished. And we can refine our methods in certain ways. So basically the psyllid LSO infecting method, if you can remember from my step A, the inoculation part, you can't really force the insects to feed on the plant. And since the bacteria is in their gut, they won't necessarily transmit if they don't feed. So that's pretty complicated. And then also, they tested for the presence of virus in the lab in Washington using ELISA, which is another method, and they said that it was infected, but when we did qPCR, we found that they weren't infected. So the different methods may have resulted in a different result, and we didn't get the same result whether they were infected or not. So, once again, the importance of this research, when we finally get it all done, like I said, by studying this interaction better, we can treat things more effectively and comply better with government guidelines. So if PVY does have an interaction with LSO, if it's a suppressor or something, we can increase or decrease the amount of it using pesticides so farmers can deal with problems early rather than later. And this is huge because I explained why LSO and PVY are so prevalent in the United States. Cool, I had a lot of fun in this project. My mentor here was amazing. She told me what to do every day, it was really fun, and I learned a lot. Thank you to Claire for opening her lab to me. Thank you very much. And then the members of the lab, Sarai, Laura, Mackenzie, Arlie, they're just great people. Thank you to Tyler for being so kind and caring, Megan and Rick for this awesome program, and thanks to all of you guys for listening and for being great peers over these 6 weeks. Any questions?
Can you go back to the results slide for set B?
Yeah, for sure.
Yeah. So compared to the other types like
This or?
No, B. Yeah. The PBYB, what's your hypothetical conclusion? Why do you think the margin of error for the PBYB is so
So basically we did 5 samples for each, right? The reason why the margin of error was so high is because even among these 5 plants, only like 3 of them were infected. So technically it shows level of infection, but the variance caused the high error bars.
So are there like more, did you do more than 5 trials for the other plants?
We have 10 samples so far, but we've only done 5 because the whole process of DNA extraction, you only do 10 to 20 samples at a time, and each time takes like 5 hours, 4 or 5 hours. Yeah.
How do you quantify the number for the level expression relative to control? Because like the y-axis has some really small numbers.
Okay, so they're really tiny amounts. So basically, my understanding is that you get the total amount of DNA or RNA present, and you compare how much of it is from the virus as compared, or bacteria, as compared to how much you get from the plant itself. So by comparing this, that's how we see there's like a really small amount of the virus or bacteria in comparison to the plant DNA or RNA. Any other questions?
On the slide where you show the map of the United States, why is the virus only concentrated on the western side of the U.S.?
To be honest, I'm not completely sure, but if it's if it's our coast, it's probably because the weather conditions here are more allowing for the LSO. Cool. Yeah.
What do those purple dots on the map signify?
Those are areas I found that are specifically concentrated in LSO. Thank you. Up next, I'd like to introduce my friend Brian Kwok.
Brian, we're about 4 minutes ahead of schedule.
I'll wait for my turn.
Okay.
We're gonna go 5 minutes. Yeah, but it's only 9 to 10 minutes.
I know. I would rather be. I guess I'm just not.
Oh yeah, sure. Watch the board. Temperature is really
Question?
Right for a lot of numbers.
the whole population.
When I download the application, compare the app.
Let's just download the file and we'll put them on that hard drive.
In the practice,
Probably work on that during,
and then
second half of
Remember the projection?
on the second half.
yeah, I think you're the only person that
I'm not.
and there's a lot of people under.
40-year-old guy has bad
I
jokes.
don't know. Like you were on the longer end. We're in agreement.
Brian, give us just another minute.
Yeah.
You'll be happy that Jimmy Buffett was on
What?
the bench for the performance. So I just
Yeah, I know that.
I didn't watch. Every time I see Jimmy.
Dude, what the heck, man?
I like your pen. I'm checking to see if your mother is online. So are the people for your lab here?
Yes.
And your mother is ready?
I'm quite sure she is.
She just told me she was.
Alright, so when you're ready.
Alright. Good morning, ladies and gentlemen. My name is Chang-Hwan Kwok, or better known as Brian Kwok, and in the Department of Chemistry, under the supervision of postdoc student Jian Wang and assistant professor Dr. Kirill Kovner, I have been exploring a new barium antimony selenite compound as a thermoelectric material. So the contents of the presentation are as follows. I'll first start off with the background information, which includes basics about thermoelectricity, and I'll talk about the barium antimony selenite compounds. And then I will talk about previous research done by the Kovner lab, and then I'll move on to the research process of my lab, which includes the methods and equipments, results, discussion and conclusion, and I'll end off the presentation with an acknowledgement slide. So first off, a few numbers. That is the average efficiency of a car. And that is the average efficiency of a car with thermoelectric materials. This 8% gap is caused by the conversion of heat energy into electrical energy for the car to reuse as fuel. So the most fundamental equation for thermoelectricity is the calculation of the figure of merit. So the figure of merit of a thermoelectric material is the square of a Seebeck coefficient divided by thermal conductivity multiplied by resistivity. So this is how a thermoelectric material works. There's a hot side and there's a cold side. And as heat transfers to the cold side, the electrons travel around the thermoelectric material, and therefore the heat conversion to electrical energy takes place. And we want thermal conductivity to be low, so we want heat to travel slowly from the hot side to the cold side, so there's more time for the electrons to move around the thermoelectric material. And we want resistivity to be low in order to have at least resistance as possible so that electrons can move as freely as possible. And you may be wondering what the Seebeck coefficient is. It is the change in voltage divided by the change in temperature. And we want the change in voltage to be great, because change in voltage becoming great means greater current, and greater current means that there's more electrons that can travel through the thermoelectric material for more conversion to take place. And the next law is the Bragg's law that many of you might know about, but I'll mention this later in my presentation when I talk about a few equipment that makes use of the Bragg's law. So about barium's antimony selenite compounds. The main focus of my research was the BA6, SP7, SE16.11 compound. And the objective of my research was to synthesize it, analyze the crystal structure, and calculate the figure of merit of this compound. And the green atom over there is the barium atom, the tanned atom is the antimony atom, and the blue atom is the selenium atom. But while talking about the 6,7, 16.11 compound, I can't forget about the BA4, SP4, SE11 compound. I'll talk about why I included this compound in my presentation later, and I'll be moving on. So there are four main structural units of the barium antimony selenite compound. First of all, there is the one-dimensional antimony 3 selenium 7 one-dimensional chain. So this chain is consisting of three SB-SE5 tetrahedral pyramids, which are connected by two vertices. And this shape is similar to the SB-SE slab found in the BA2, SB2, SE5 compound that was previously researched in the Kovner lab. And similarly, the one-dimensional barium-2 selenium chain is also found in the BA2, SB2, SE5 compound. Next up, the one-dimensional antimony selenium 3 chain is a tetrahedral chain that consists of SB-SE4 tetrahedral chains that are connected by two vertices. And lastly, there is the two-dimensional antimony 4 selenium 6 layer. And this is connected by condensing alternating SB-SE5 square pyramidal chains and SB-SE3 pyramidal chains. And they're connected by connecting all the vertices of the chains except for the apical vortex of the SB-SE5 pyramid. So previous research done by the Kovner lab. So the Kovner has researched many compounds such as clathrates, zinc antimonides, copper phosphides, and other barium antimony selenite compounds. On the left is an example of a clathrate compound, and on the right is an example of a copper phosphide compound. So the research process. So here's how it goes. I put the elements in a stoichiometric ratio of 6, 7, and 16.11 respectively, and I put them in a vacuum tube so that the oxygen doesn't react with the elements while synthesizing. And after putting those in a tube, I labeled two compounds, BK001 and BK002, and I put those two compounds in a furnace where I synthesized for over a week. So after the first week, I took out the BK001 compound, and according to X-ray powder diffraction results, it was synthesized as a 4,4,11 compound, not a 6, 7, 16.11 compound as I had desired. And a week later, I took out the BK002 compound, and unfortunately this was also synthesized as the 4,4,11 compound. So we decided to improvise and put the BK002 compound that was already synthesized into a spark plasma sintering device, where we adjusted the temperature and pressure of the surrounding of the compound and decided to make it stay there for 20 minutes. But unfortunately, the BK003 compound that was synthesized was also 4,4,11. So For sampling of the elements, we used an argon-filled glove box. And we needed to use an argon-filled glove box because of the toxicity and the carcinogenic properties of the antimony elements. And as I mentioned before, there are two main equipments for synthesis. First, there's a spark plasma sintering device, where the temperature and atmosphere of the compound is adjusted so that the reaction takes place much more quickly. But then the disadvantage of this device is that it is unsafe, as seen by the selenium element dissolving, evaporating during the synthesis. But the furnace is much more safe. It goes through thorough annealing, which takes a process of a week. But as I've just mentioned, a week is too long for a 6-week camp. The equipment that we use for X-ray powder diffraction is the Rigaku 600 Miniflex X-ray powder diffraction. So I mentioned this before, but this machine makes use of the Bragg's law, and it uses the angle of reflection and incidence of the X-ray light, and it calculates the intensity of the X-ray light that is bounced off the compound, and it uses that to determine the crystal structure and the crystal lattice planes of the compound, which helps identify the compound. So the graphs of the X-ray powder diffraction are as shows. The y-axis is the intensity of the X-ray light that is reflected off the compound, and the x-axis is the angle of incidence of the X-ray, which is shone upon the compound. The top graph is the X-ray diffraction results of the 4,4,11 compound. The middle graph is the results for the BK001, and the bottom is the theoretical result of a 6,7,16.11 compound. As you can see, the intensity peaks of the BK001 compound are much more similar to that of the BA4, SB4, SE11 compound than it is of the 6,7,16.11 compound. And similar results were shown for the BK002 and BK003 experimental compounds. You can see that there are intensity peaks at the initial angles of the 6,7,16.11 compound, which the 4,4,11 and BK002 compound does not exhibit. So the last equipment that we had to use for our experiment was the physical properties measurement system, which measures the thermoelectric properties of my compound and helps me calculate the figure of merit by determining the thermal conductivity, resistivity, and the Seebeck coefficient. However, during the middle of this program, this machine broke down, and we were unable to measure the thermoelectric properties. So why did the 4,4,11 compound become synthesized? Well, the first reason that we concluded was the stoichiometrical ratio, the similarity of the stoichiometrical ratio of the two compounds. Once recalculating the stoichiometrical ratio, the 4,4,11 compound can be restated with a ratio of 6, 6, 16.5, which is very similar to the ratio of 6, 7, 16.11, except for the one antimony atom. Another reason is the similarity in structure between the two compounds. I have mentioned before that there are 4 main structures that make up a barium antimony selenide compound. Both of these compounds exhibit those properties of the Ba-Se slabs and the antimony Se one-dimensional and two-dimensional slabs. But then, why did the 6, 7, 16.11 compound not get synthesized? We concluded that the reason of that is the unbalanced structure of the compound. If you can see the electrical configuration here, it shows that the Ba6, Sp7, Se16.11 compound has a net ionic charge of 0.8 plus. And on the other hand, the 4,4,11 compound has a net ionic charge of 0. So we concluded that the 4,4,11 compound is much more stable than that of the 6, 7, 16.11 compound, and therefore the reaction moved over and transitioned into the 4,4,11 compound during the synthesis. So the future applications of the barium antimony selenide compound. So previous barium antimony selenide compounds were known to exhibit low thermal conductivity. And low thermal conductivity is good for the aforementioned increasing efficiency of cars' engines, because low thermal conductivity can mean there's more time for the heat to move on to the cold side, so that there is more time for the electrons to travel through the thermoelectric material. So I would like to acknowledge a few people for my research. First of all, I would like to thank postdoctorate student Jian Wang for helping me throughout the entire camp and dealing with my mistakes. And I would like to also thank Assistant Professor Carol Kovner for letting me use the lab and have a great 6 weeks of research. And I would like to thank all the YSP staff members, Jeremy, Tyler, Anne, Corinne, Stephen, and of course the directors Rick and Megan for organizing such a well-made camp and letting me have a great experience. And if you're watching through the camera, Mom and Dad, thanks for giving me this 6 weeks of learning and great time to spend with friends. And finally, I'd like to thank all of you and King, because, yeah. Here are my references, and thank you for listening. Any questions? Jane?
Can you go back to the slide for the structure of the 1611 compound?
Okay. Yeah.
So what are the white spaces
So the white spaces are unoccupied regions of the atoms. And the unoccupied regions are spaces that are not filled up with certain atoms. So that is the blue part, which I said before is the selenium part. So this part is not filled, and therefore you can see that's the reason why there are so many selenium atoms, because it's not filled and it has to make up for the number in other places otherwise. Jackie.
Why is your 6,7,16.11 compound, like, why do you think it'd be better for thermoelectric purposes?
Can you repeat the question?
Like, if you mentioned in the beginning that you wanted to synthesize compounds for, like, better thermoelectric, like, properties so that you can have a more efficient engine, but why do you think this is better than, like, say, the 4,4,11 compound?
So the 6,7,16.11 compound has actually a much more complex structure than the 4,4,11 compound. And having a more complex structure means that it can have a lot more physical properties that we don't know of. So it's very promising. But any lab around the world has yet to synthesize this compound. It's never been synthesized before. Even before I came to this camp, for 2 months, my mentor said he could synthesize it. So we have yet to synthesize it. Renee?
Oh, why did you choose to create the 6,7,16.11 rather than some other compound?
The barium antimony selenite compound, as I said before, is highly promising, and it was really, I thought it would be really interesting, and it sounds cool. Look at that. Complex. Yeah. David?
What would be a high figure of merit?
So a high figure of merit, so starting from 1, above 1, you can use it for any thermoelectric applications. Over 2, that's really good, and the highest recorded thermoelectric figure of merit is 2.6, 2.7, and over 3, that's unheard of. That's never been synthesized before. Yeah. Okay.
So why do you think it's so hard to synthesize?
I said this in the discussion slide, but I said this because I said it's hard to synthesize because of the unbalanced structure and the complex structure of the 6,7,16.11 compound. I said that it has a net ionic charge of 0.8 plus, which makes it very unstable compared to the 4,4,11 compound, which is very stable compared to this compound. Listen more next time. Henry?
You said that these compounds are both toxic and carcinogenic. So
Not the compound, the antimony.
Oh, okay.
Yeah. Jack?
So could this material or structure be used to replace, say, an alternator?
I don't know what it is.
Like it's the part of the engine that converts into electricity off of engine power. Could you replace that?
I'm not sure about replacing, but I guess we could use it along with it.
Augment?
Yeah. Sure. Sunny?
So how would this compound be applied to the engine? Do you know about that?
I'm not sure about that.
Okay.
I'm not quite sure about this. Sorry. David?
Why is there like this and 16.1?
Because if we make it to a whole number, that's long. Very, very long. Try it. That would be near like, it would be like B 600 and 700.
Oh, I see.
And so, yeah.
Oh.
Any other questions? Thank you. Next time, I would like to introduce my colleague and friend, David Wong.
David, is everybody who's coming to see you here?
I think so.
Okay, because we're about 2 minutes early. Do you have somebody that's watching at home?
I don't think.
Okay, so if you'd like to begin, you can.
Probably like that. That's not it wasn't too fast.
So hello everyone, my name is David Wang, and for the past 6 weeks I've been working at the Korf Lab at the UC Davis Genome Center, and I've been working under the mentorship of Dr. Ian Korf and Hannah Lyman, and what my project is aiming to do is try to distinguish C. elegans chromosomes using amplicon-sized polymorphisms. So C. elegans, or Cnr hepatitis elegans, they are a free-living transparent nematode found in temperate soil environments. They're easily cultured in a lab, and they have a relatively short life cycle of 3 days, making them an ideal research animal. In addition, 65% of human disease genes have homologs on its genome, which further strengthens its virtues as a lab research organism. So some previous research done on these organisms include being the first multicellular organism to have its entire genome sequenced. As you can see, 6 chromosomes on C. elegans, 1, 2, 3, 4, 5, and X. In addition, its reproductive processes such as chromosomal mechanisms during meiosis and also sex determination have been thoroughly researched. They have found 3 dose-sensitive sites on the X chromosomes, which all act together in determining sex. And in the figure above, we have chromosome movement and restructuring during meiosis. So some current research done at UC Davis is Dr. Frank McNally in the Department of Molecular and Cellular Biology. He's trying to investigate chromosomal loss in Aneuploid C. elegans. So surprisingly, a tetraploid C. elegans worm, which means has 4 sets of each homologous chromosome, has shown to be viable and display normal characteristics save for their enlarged size. So if we take a tetraploid C. elegans worm, it's from the N2 Bristol strain, and we made it with a normal diploid worm called strain 1. They both undergo meiosis, which the number of chromosomes have, and this produces a triploid C. elegans individual, with 2 homologous chromosomes from the 4N and 1 from the 2N. Now what the research is trying to study is the frequencies for each of these possible gametes that the triploid individual could produce. What chromosomes does it lose and what chromosomes does it keep? So in order to test this, we made it with a 3rd strain, also a diploid, have it undergo meiosis, and once again there's 4 possible combinations for this 3rd generation C. elegans. The problem with this is that there's currently no cost-effective way of distinguishing these homologous chromosomes, which chromosome which came from which parent. So some possible methods that were considered before are genome sequencing. Basically, we just sequence each chromosome before and after reproduction to see which ones they kept and which ones they lost. Unfortunately, this is a very expensive process and also very tedious. In addition, we've considered fluorescence in situ hybridization, which is a process where they use antibody tagged DNA probes, make them pair up with complementary sequences on certain chromosome numbers, and then they'll fluoresce and be visible under a microscope. So this is a human karyotype, which a number of chromosomes are fluoresced using this method. However, this is also a very tedious process since you need 6 simultaneous dyes at the same time in order to distinguish what chromosomes came from which parent. So our proposed method, we want to develop a cheap, efficient way of distinguishing these homologous chromosomes. So we hypothesized that there will be a region of around 1,200 base pairs on the same place in each of these 3 strains, as mentioned before. The N2 strain, which is a tetraploid, would have no mutations. The strain 1 would have a indel or insertion deletion polymorphism, and strain 2 would have a indel of significantly different size. So if we run this, if we amplify these 3 regions through PCR and run it through electrophoresis, we would expect it to be distinguishable through our method. Our method uses universal tools in biological research, so it would be both cheap and efficient. So our main objective was just to find strain pairs. Since the N2 Bristol strain has no mutations relative to the other ones, we want to find strain pairs instead of strain triplets with these pairs on the most number of chromosomes possible, meaning we want it to be ideally on all 6 chromosomes, so we can just use those strains to test aneuploid loss. So we retrieved the polymorphism dataset reference to the N2 Bristol strain from WormBase, which is like an online database of worm genomes and annotations. Each line had this information, which we used to determine if it was a polymorphism pair, and we implemented constraints on these values. So the factors which influenced these constraints were sample size, the size of the DNA, the voltage of the gel electrophoresis, and the ionic strength of the buffer. We determined that ideally there would be these polymorphisms would be around 50 to 900 base pairs in length. The range would be less than 1,200 base pairs, as seen before, and the length difference of the 2 mutations will be greater than 50, so it will be distinguishable on gel electrophoresis. So we iterated over the dataset, which means we just went over each line, and there were like millions of lines of these polymorphisms, and we checked whether pairs would meet these constraints. After each polymorphism, there's also a list of strains on which it existed. So if we found a match, we could take the strains from the first and strains from the second and make pairs with each one. And another thing we had to consider is that we need mating plugs in these strains in order to visibly confirm mating in Aneuploid C. elegans under a dissecting microscope. So eliminated strains without mating pairs. Again, after we got all these strain pairs, we iterated over that to see which ones had the most polymorphism pairs on the most chromosomes. Another thing we had to consider is primer design. So in order to isolate these regions of indels, we had to design unique primers surrounding the region of insertion deletions. So we use Shannon's entropy, which is returns a value between 0 and 2, with a higher value being more complex, so that means just less repeat regions, which we don't want. We want sequences, unique sequences, which we can use a primer to anneal the splice of DNA. So it basically screens for high complexity regions and eliminates low ones. So for our results, for polymorphisms under 1,000 base pairs, we found these many insertions and deletions. The average insertion length was around 3.42 base pairs, and the average deletion length was around 57.2 base pairs, which is like a significant difference. In total, we found 981 of those possible polymorphism pairs meeting all constraints. And this is a really exciting part. We actually found 6 strain pairs, which with at least 2 of these polymorphism pairs on each chromosomes. So we can theoretically use these 2 polymorphism pairs, strain pairs, to test chromosomal loss in Aneuploid C. elegans. So here's just a figure of where all these polymorphism pairs are on each of the strain pair. As you can see, the first 4 strain pairs all have the same 2 polymorphism pairs on chromosome 3. So many of these mutations are conserved between strains. The reason why we would want to know the location of these polymorphisms is because during recombination, they may trade places and falsify data. So if we just revisit our method once again, we just have regions of around 1,200 base pairs on 3 strains with distinguishable polymorphisms that we can amplify and distinguish through PCR gel electrophoresis. So this is an output of my program, which after iteration, I modified it so it would be more flexible. Instead of looking at 4 or more chromosomes, I did 3 or more. So this is the strain pair ED3049 and AB3. So I don't know if you can read this, but the ED3049 has a deletion length of 59 within this specific region, and the AB3 has a deletion of 146, and the N2 strain has no deletions. So we would expect from our method that the N2 strain would be the biggest sample size, the AB3 would be the smallest, and ED3049 would be in the middle. So when they ran this in the McNally lab, we actually see a significant difference in these predicted product sizes. So this suggests that our method is a viable method for distinguishing chromosomes. So to summarize, 6 ideal strains were found. All polymorphisms were deletions, which meant that there were no insertion polymorphisms that met our constraints. Lab work deemed our method as viable, and testing on those 6 strain pairs I found will be done in the next few days or weeks, because I found the strain pairs around 2 weeks ago, and they ordered them, and apparently they're still shipping, so I won't be able to see results until after YSP. Okay, so there's multiple directions which we can take this research. First off, we can obviously now study chromosomal loss in Aneuploid C. elegans. As I mentioned before, we can study recombination during meiosis, so we can pick those polymorphism pairs that are the least likely to be switched places on homologous chromosomes. We found a discrepancy between insertions and deletions, so we can research how exactly is a genome mutated, and somehow insertions and deletions counteract each other. And also, this method can be used to distinguish chromosomes in other organisms and model systems, which can be used for health, biomedical, and genetic research. So here are my references. I'd like to thank Dr. Ian Korf and Hannah Lyman for welcoming me into our lab, and also Dr. Korf for going halfway into my project to see if it could actually work before giving it to me. Frank McNally and Elizabeth Vargas for the laboratory work, providing that gel electrophoresis. Robbie Dankar and my roommate Jack Ustra for code troubleshooting and just for being an awesome roommate. Kim Trenton for counseling the program. Rick Pomeroy and Megan McKenzie for the hard work in organizing the Young Scholars program. My family, my mom is here today for motivating me to pursue what I love in life, and to this big family sitting in front of me right now that we all become over the past 6 weeks. Thank you.
Question. Is there, with the C. elegans, is there like any specific reason why you chose it? I mean, like, is there any significance? Like,
Well, we want to research C. elegans because they are a very versatile and potentially very valuable research organism in the lab. And they could eventually maybe replace mice since they're so easily culturable and so inexpensive and efficient.
I think there's a slide where you had like a lot of colored chromosomes. What were like the numbers labeled in the wall?
Numbers?
Like
This one?
Yeah. Like what are the CV4?
Oh, these are just the names of the strain.
Okay.
Yeah, the strain, like, the first one would be the first strain pair name, and the second one would be the second strain pair name.
Okay.
They will all be researched with the N2 Bristol strain.
And with Roman numerals, goes from 1 to 5 and then jumps to 10.
Oh, it's just the X chromosome.
Oh,
yeah.
Okay. That makes more sense.
Good eyes, Jack.
Okay, thank you guys. I'd like to present my colleague, friend, and lab mate, Richard Yu.
Richard, we're running about 4 minutes ahead. Are the people who are here to see your presentation, are they here already?
Yeah.
Okay, and is anybody watching you online?
No.
Okay, then we can go ahead and move forward if you'd like.
Oh,
Can
okay.
you close David's tab when you get up there also?
Yeah.
Thank you.
So we
Whenever
good?
you're ready.
Start? Okay. Okay, good morning everyone. My name is Richard Yu and I'm also working in the Corf lab, the same lab that David's working in, but our projects are quite different. Very different actually. And so my project is on unusually high conservation between the genomes of the nematode C. elegans, the worm that David introduced you to, and another worm called C. brixi. So a human, a rat, and a mouse, approximately 80 million years ago, they shared a common ancestor. And over all these years, many mutations should occur due to genetic drift. So it was a shock, however, when researchers in the early 2000s saw regions between these three genomes that are 100% identical. And if we zoom out of this, we can see that these regions are 200 or more nucleotides in length. And there weren't just a few of these, there were hundreds of these. There were 481 of these to be exact, in between the human, rat, and mouse. And they were named ultra-conserved elements. They were first defined between the human, rat, and mouse as being 200 plus base pairs in length. But they've come to take in have a more flexible definition. For example, in this year, Drosophila UCEs were defined as 50 plus nucleotides in length. There have been some functions proposed for UCEs, such as epigenetic and transcriptional regulation, but they have not been definitively proven. In mice, UCEs have been deleted, but no abnormalities were observed, which proves that UCEs are possibly not essential for an organism's functions. In vertebrates and insects, UCEs are mostly located on introns, intergenic regions, and intron-exon junctions. And in mammal UCEs and plant ULEs, which are UCE-like elements, it's basically just a different name for plants. There was a drop in AT content, which basically implies an increase in GC content. So all this background information I'm telling you now is important for what I will tell you later on about my findings, our findings. right now we've been talking about 100% DNA conservation, but then we wondered, what about 99% or 98 or 97% DNA conservation? That's also very strangely high levels of conservation after, let's say, 80 million years, so many years, right? So we shifted our focus from ultra-conserved elements and 100% DNA conservation to unusually high DNA conservation. And we coined a term for regions of DNA in this area of unusually high DNA conservation. So we called them peculiarly conserved elements, PCEs. So we asked ourselves, what are the properties of PCEs? So we categorize UCEs as basically a subset of PCEs, the 100% conservation PCE, basically. PCEs basically just incorporate UCEs and the other regions of DNA that have lower than 100% nucleotide conservation. So we used C. elegans and C. brixi to investigate this question of what are the properties of PCEs. So we use C. elegans for the reasons that David mentioned, and we use C. brixi because they are pretty actually evolutionarily distant from C. elegans. So we can get kind of the same nature between humans, mice, and rats. That's what we kind of were hoping to achieve. So that if there were PCEs or UCEs between these two worms, they actually would mean something significant, rather than just due to them being very evolutionarily similar. Also, the C. brixi genome is relatively intact, so that's why we chose. That's another reason why we chose it. So we use a Mac computer. You might be familiar with that for basically all our work. And the Mac operating system, OS X, now known as Mac OS, it actually has a Unix-based foundation. Unix is an operating system, and we can use Unix commands on the Mac by using an application called Terminal. This is what the icon looks like, the black icon. And if you open it, you can type in Unix commands on it. In conjunction with Unix, we use a programming language called Perl. And using Perl, we can write scripts. We could write scripts and run them on Terminal. So we asked ourselves, what is considered peculiar, right? What would be considered peculiar conservation? The difference between typical, normal conservation and strange, peculiar conservation. So we've put our first step of our project to determine the peculiar threshold. As you see, we're going along, we're asking questions and going along as we ask questions. So this is a pretty discovery-based project. So here's an overview of what we did to give you a sense at first of what we did. So first we look at the amino acid sequences with 100%, not the nucleotide sequences, the amino acid sequences, because that way we can see the nucleotide sequences relative to the amino acid sequences and determine what is typical for 100% amino acid conservation. Now if we look at the corresponding nucleotide sequences, we look at their percent identities, and then we found the mean and the standard deviation of those percent identities. And then we took 2 standard deviations above the mean to get our minimum peculiar conservation percentage. I know some other projects have also done standard deviation and 2 standard deviations, I think. So here are the specifics now. We downloaded the protein sequence and coding sequence for each worm from Wormbase, the database that David showed you. We created a BLAST-P file, which is basically an alignment file of the protein sequences. P basically means protein sequence. So we wrote a Perl script using this alignments file, this BLAST-P file, and this basically is a graph of the output, which gives us a distribution of the number of alignments for various amino acid identities. This gives us a sense of the distribution, basically. And to actually get the mean and the standard deviation, we wrote another script that uses BLAST-P and the coding DNA sequence to give us the corresponding nucleotide percent identities, as I was talking about earlier. And as you can see, we chose 99 to 100% amino acid identity. It says 99, but actually means 99 is the starting point up to 100. So 99% to 100%. The reason we chose 99 to 100 is to account for potential experimental error that may have occurred during the sequencing of the genomes. So the mean is 87.74. We did 2 times 4.5, which is the standard deviation, added it to the mean, and we got truncated the decimal, and we got 96% as our nucleotide conservation peculiar threshold. So we're done with step 1. Yay! So what do we do next, right? So next, we have to determine a length minimum for the PCEs. We needed to find the number of PCEs. We needed to determine we chose to determine the regions that the C. elegans PCEs overlap because, as I told you earlier from the background, I told you about the overlapping regions in other organisms. So here we're trying to see, are there differences in the PCEs versus the UCEs in other organisms? And also, we further investigated the properties of PCEs, and I'll show you the specifics in the results. So to address all these steps, we basically kind of addressed them together. We downloaded from Wormbase the genome sequences and the annotation file. An annotation file basically tells you key regions on a genome and gives you the coordinates for things like introns, exons, and things like that. We created a BLAST-N file. The N stands for nucleotide. So instead of BLAST-P, this is BLAST-N, which means it's aligning nucleotides instead of protein sequences. And this is basically part of what it looks like, just to give you a sense. It's probably very small and you can't really read it, but this is just to give you a sense of what it looks like. So we wrote some other scripts to address the other steps. That's all I'll say. Okay, so for the results, we chose 96% as our nucleotide conservation, as I said, and our length minimum was 100 nucleotides that we set, because the minimum length was 89 nucleotides, and the average was around 145 for the PCEs. So 89 was the minimum length for the PCE candidates. So we chose 100% because we felt like it would not be too restrictive, but it would also be a good length minimum. We found 385 PCEs. Most of them are in coding regions. As you can see, 69.9% are in coding regions. The rest are non-coding. And here's a different perspective with the regions that we found. Most of them are located on exons and intron-exon junctions. As I said earlier in the background, there weren't UCEs that were mainly located on exons, so this was an interesting finding. Most of them also shared coordinates. Most of the PC also shared coordinates with mRNAs. As you can see, most of them shared coordinates with mRNAs, and some of them were combinations of various RNAs, and they still included mRNAs, most of them. Another result that we found was that PCs had greater GC content than 1,000 arbitrarily selected introns, exons, and CDS segments from the C. elegans genome. As you can see, it's 50.5% for the GC content for PCEs, which is notably higher than the others, as you can see. And this again takes us back to earlier when we were talking about the drop in AT content, which suggests a rise in GC content. I'll elaborate that more later. We found 5 intergenic PCEs, and all of them lie on the X chromosome, which is very strange. And we kind of have speculations, which I'll talk to you later about, what we speculate for why they are all located on the X chromosome. So basically, here's you can see here on the very corner over there near where it says results, it says X, which means X chromosome. And here's an intergenic region, as you can see over here. And it is indeed an intergenic region. This is one of the intergenic PCEs that we put on the Wormbase genome browser to let you see it pictorially. So our findings were that there was a rise in GC content for mammal UCEs. There was already we know that there's a rise in GC content in mammal UCEs and plant ULEs, and here we found that there's also a rise in GC content. And this suggests that there's similarities in unusually high conservation between all these organisms, and also that we are able to generalize ultra conservation to peculiar conservation. So we can view these different UCEs, ULEs, and PCEs similarly. Another thing is that, as I said earlier, there are many PCEs located on exons in C. elegans, while for vertebrates and insects, many of them were located on introns intergenic regions, but both of them are located on intron-exon junctions. But there's a difference in that many are located on exons, so then there are still differences in unusually high conservation between all these organisms. So here are the significance and implications of my findings, our findings. PCEs may carry over, our findings here may carry over to vertebrates, even though our findings here were in the invertebrate nematodes. So perhaps these tests can be done in vertebrates to see if the results are the same or are there differences, and then those can be analyzed. Also, we speculate that all the 5 intergenic PCs being on the X chromosome could be due to something called dosage compensation. And dosage compensation is basically autosomal regulation of expression of sex chromosomes. As you probably know, in a female with 2 X chromosomes, only one of the chromosomes are actually expressed, and this is due to things in the autosomal chromosomes that regulate that. So that's basically what dosage compensation is. It's just a speculation. Further research can be done on this for more information. And our findings also can give more information about nematode genomes. So scientists, researchers working in studying nematode genomes can gain further insight based on these findings. Further research, here are some further research that we suggested. So we analyzed PCEs on C. elegans, but we didn't analyze them on C. brixi. The PCEs are between both C. elegans and C. brixi, but we only analyze them on C. elegans. So further research can analyze them on C. brixi. And another thing is that we can test if PCs are orthologous. We don't have time in 6 weeks to test all these things. We wish we could, but so we put them in future research. So orthologous, basically what we mean by this is if a PCE is located on an exon in one worm, is it also located on an exon in another worm? And if it's located on an intron in one worm, is it also located on an intron in another worm? Same goes with intron-exon junctions in intergenic regions. Another thing is we can test are there certain amino acids that appear more frequently in PCEs, and are there certain functional groups that may appear more frequently? And maybe this could guide us to determining the functions of PCEs and UCEs. So I'd like to acknowledge Dr. Ian Korf for being an amazing mentor, an amazing, awesome mentor. Hannah Lyman for being such a great supervisor, for providing so much help. Ravi Denkadar for helping in Unix and Perl. And Dr. Francis Manali for briefly giving sharing his views on the scope of this project. And Dr. Pomeroy and Megan McKenzie for making all this possible through the Young Scholars Program. Kim Trenton for being my counselor for most of the program, and Megan again for now being my counselor. And all other YSP counselors for just being there for me and amazing. Thank you, and all the other YSP students and my family, my mom, dad, and sister are right there. Thank you very much for all your love, care, and support. So these are my references.
I know you said you don't know for sure, but is there any hypothesized purpose of the PCEs right now?
Well, for example, one thing I said was possibly for dosage compensation. Maybe the intergenic PCEs might play a role in dosage compensation. Maybe they are important factors, but we haven't gotten time to test them yet, right?
Is there a significance to like the intron-exon junctions? Like, do you have a hypothesis as to why they're so conserved?
Well, we do not have a hypothesis right now for why intron-exon junctions are very conserved, but future research can take this information and see how they can apply it possibly to functions of PCEs.
I'm not sure if I have the numbers right, but I believe you said there are 481 ultra-conserved elements, and
between the rat, mouse, and human.
Yeah, and there's less than 400 for the PCEs. Do you know why there's more for ultra-conserved rather than peculiar?
Yeah, if we look at UCEs on on the two worms, there were actually only like 2 UCDs. And that's because the genomes for the 2 worms are much smaller than the genomes between like human, mice, and rats. And so you can't really view them like identically in that way. So we also the number of alignments, number of PCs can kind of be flexible if we change the parameters of the blast end file. But this is just based on our parameters. We got 385 PCEs. So that's how it is. Yeah.
And
Last question.
for between humans, mice, and rats, do you know if there's more UCs than PCEs, or what's like the ratio kind of thing going on?
Can you say that again?
Do you know if there's more ultra-conserved versus peculiar conserved elements between humans, mice, and rats? Or is this yet to be?
There are definitely more ultra-conserved elements because, and there are definitely more peculiarly conserved elements because ultra-conserved elements we are defining as a subset of these peculiarly conserved elements. As ultra-conserved elements we're defining as 100% nucleotide conservation, and PCEs we can say, for example, like 90-something, 96 for here as our conservation. So there are definitely going to be more segments for PCEs.
Thank you.
Next up, I would like to introduce my colleague and friend, Eileen Toe.
Hello everyone, I am Eileen Toe and I'm currently an incoming senior at Saratoga High School, and for the past 6 weeks I've been working in the Zerbe lab in the plant biology department under the mentorship and supervision of graduate student Katie Murphy and Dr. Philip Zerbe. And for the past 6 weeks I've been focusing in on the enzyme activity that is involved in maize stressed events. So a bit of background information on maize. Maize is commonly known as corn to some of us, and maize, right now it is serving as the largest or the most harvested crop in the United States. It serves as a source of food, livestock feed, and biofuel, and overall it is a major resource in our global food security. However, in this past century, there have been a lot of rapid climate changes that have been occurring, and that is because of the increase in atmospheric or greenhouse gases such as carbon dioxide, and global warming has been intensifying droughts across the world in the past century. And our maize cropping systems, they rely heavily on natural precipitation, and because of these droughts, there have been so many significant crop losses, and there's an increase in pest and pathogen damage that make that even worse. And in order to make matters worse, I would say, our global food production will need to rise at least 50% in the next 40 years in order to meet the needs of our expanding population. So in order to respond to these biotic and abiotic stresses, maize, it produces a series of, or an array of specialized metabolites, and these metabolites are specialized in order to execute physiological functions in the plant. And of these metabolites, diterpenes constitute the largest and the most diverse group. And they're polycyclic 20-carbon isoprenoids, or unsaturated hydrocarbons, and they are found in the essential oils of the plant. And what the metabolites do is that they create different diterpene structures in the plant. So according to past research, all of the plant diterpenoids come from a common substrate, which is geranyl-geranyl diphosphate, or GGPP for short. And the diterpene synthase enzymes work together in order to generate compounds. And in angiosperms or flowering plants, 2 diterpene synthase enzymes, one of them has to be a class 1 enzyme and the other is a class 2 enzyme. They function sequentially in order to create different diterpene hydrocarbon scaffold structures. And the class 1 enzymes use up the class 2 enzyme products in order to generate these compounds. So in GGPP's case specifically, there is a class 2 enzyme called anther-ER2, or AN2, and it uses up the GGPP substrate as part of the plant's defense process and creates N-copylyl diphosphate, or N-CPP. And recently the Zerbe lab identified 2 diterpene synthase enzymes which are class 1 enzymes, and they are choline synthase 4, or KS4, and choline synthase 2, or KS2. And KS4 and KS2, they work to convert the N-CPP substrate and create two different compounds. So for KS4, dolabridine, which was a previously unknown diterpene, it creates, dolabridine enhances the drought tolerance in the maize root tissues. And on the other hand, KS2, it uses up the N-CPP substrate and creates N-isocorine. And N-isocorine is a precursor of what are known as coralexins. Coralexins, they provide protection in response to biotic or abiotic stresses such as insects or pests or pathogens or droughts. And it accumulates in the roots and it provides protection for the plant. And what we've been doing is that we've been investigating the structural and the functional differences between these two proteins. They're so closely related in that they utilize the same substrate, but they're different in that they create these different compounds. And what I've been doing is that I used site-directed mutagenesis of the KS2 protein so that I can see how the mutations in the genes can affect the induced metabolites. And in a broader sense, this can help us identify the amino acid residue function or the role in enzyme activity of maize. And it can also give us more of a sense of the overall plant diterpene diversity in maize, as well as how the plants have evolved over time through adaptive mutated processes, such as neofunctionalization or duplication, in order to generate these type of compounds. So these are the methods that I followed. I first started with sequence alignments, which compared the amino acid sequences between KS2 and KS4. And in order to actually understand how the amino acid residues function in enzyme activity, we took more of a visual approach with 3D structures with homology modeling. And from homology modeling, we were able to identify key residues that we could mutate in mutagenesis, which is our next step. And from mutagenesis, we had mutated plasmids. And these plasmids, they were grown in E. coli colonies, and those colonies expressed protein, which is our next step. And from that, we were able to get induced metabolites to run on our gas chromatography, mass spectrometry, or GCMS for short, machine for further analysis and our results. So this is my first step, which is sequence alignments. I basically got the amino acid sequences between KS4 and KS2 and compared them. And in the figure above, you can see that the yellow shaded areas represent the similarities between the KS2 and KS4, while the gray areas represent the disparities. And we found that just from this, there's 57% similarity between KS2 and KS4 in terms of amino acid sequences. And this is only consistent with the fact that they do produce different compounds. So we wanted to further investigate what the actual role and the function of the amino acid residues were in enzyme activity. So that brings us to our next step, which is homology modeling. And that was visualizing the KS2 protein. So for homology modeling, we got templates from the RCSB protein data bank, and we got this template that was structurally related to our protein, and we molded our protein to this template. And from this template and this model, we were able to identify residues that were located in the active site that we could use for further mutagenesis. And this is a close-up of the model. And the yellow and orange structures represent the ligand structure, and the pink, blue, red, white stick structures represent the residues that we tested out. And we used PyMol, which is the software for visualization and ligand docking. And based off of the residues that we found, we were able to design primers and order them for our mutagenesis. And that is our next step. So for mutagenesis, we used PCR, our polymerase chain reactions, and we amplified the DNA in these plasmids that are E. coli colonies. And the E. coli colonies are grown on selective plates of LB agar that grow the E. coli. And from that, we were able to extract DNA through a process called mini-prepping. And we grew them in the DH5 alpha cells, and the extracted DNA was able to be shipped off to a company called Kintara Bio, which sent us back sequencing results so that we could actually tell if our mutation was successful or not. And from that, we were able to find that the isoleucine to threonine at site 644 and the serine to proline at site 509 were successful for mutagenesis. And those were the 2 residues out of the H residues that we tested that were actually successful. And from those 2, we chose serine at site 509, or I will now be calling it S509P for short, for further co-expression assays. Oh, okay. So our next step is protein expression. So we transformed the DNA into the colonies that are transformed into the coli colonies again, and we grew them on these plates like this, and the speckled dots are the coli colonies. And from these plates, we grew them, and we swapped 2 colonies from each plate, and we grew them into these vials that we would be running on the GC for analysis. And the cultures, they were induced for protein expression and metabolite production. So this is what our GCMS looks like. And on the left, there are the GCMS vials. And we added hexane to each of these vials to create a hexane layer with the extracted metabolites. And these metabolites were then stuck in this hexane layer. And on the right of the GCMS, there is a needle that dips into this layer and provides us with our results. So this is what comes out of a typical GCMS. And we had a wild type or a control enzyme that we can compare the standards and the products that are formed from it with the results from our mutant. So the first is our wild type gas chromatogram. And in order to read these gas chromatograms, you have to look at the peaks, and each peak represents a product that is formed. So in this case, our peak is N-isoprene, and that is our KS2 product. So if you focus in and zoom in on the peak, then the mass spectrum, or basically all of the masses from the sample that is tested, is shown below. So this is the results these are the results that came out of our GCMS for our mutant S509P. And overall, there has been a reduced enzyme activity that has been shown, and that is because there is a reduced formation of N-isoprene, the KS2 product, in the gas chromatogram of S509P. And also there's a presence of NCPP, which is actually the substrate that KS2 uses in order to convert it to N-isoprene. And NCPP isn't present in the wild-type gas chromatogram. Also, there are 2 new products, which are N-corine and, so far as of now, an unidentified diterpene. So the significance of these results is that we were able to find something, specifically serine at site 509, that we believe has a critical role in enzyme activity and specificity. Serine 509 wasn't able to convert enough of the NCPP into N-isoprene, so that's why both of them are present in the gas chromatogram. And also we believe that it has a potential to control the position of the substrate in the active site. And from this, we were able to be one step closer into actually identifying what the function and the role of the amino acids are in the enzymes, and also get a better sense of the plant diterpene diversity in maize and how plants have evolved over time to create these compounds. And in the future, this can help us lead to find a way to discover, to develop secondary metabolic pathways that can help us create more of a new crop management system with more resilient crops with a higher stress tolerance and resistance. And these are my references. And for acknowledgements, I would like to thank Dr. Philip Zerbe and graduate student Katie Murphy for welcoming me into the lab and helping me with everything possible, from the drafts that was sent in at 3 a.m. of my paper to any failed reaction. Also, my counselor, Kim Trenton, she's not here today, but also Megan McKenzie for also taking her place and being my counselor. And Dr. Rick Pomeroy for giving me the opportunity of a lifetime to be here at the Young Scholars Program. And also everyone from YSP for being my family for the past 6 weeks. And also my real family who are in the back, as well as my chem teacher, Mr. Cruiser, who is also in the back, and he has helped me open my eyes into the world of science. Thank you. So are there any questions? Sabrina?
So why did you specifically use E. coli?
So we use E. coli because we thought it was the best system in order to induce these metabolites. It's able to store the DNA in a stable environment, and also you can amplify DNA in a more of an efficient process than if we actually did it manually. Renee?
When you chose colonies to grow, did you do anything to verify that the colonies you selected had what you were looking for?
That's why we sent them off to Kintara Bio, so that they can give us the sequencing results, and from that we can see if there was actually a successful mutation. Some of the colonies actually had like insertions or contaminants, and from that, that's why we were able to narrow it down from 8 residues to 2 that we could actually use for further co-expression. Thank you.
We'll be back at 10:30 with Sarah.
Oh,
been working with the Department of Plant Sciences under the mentorship of Dr. Ken Shackle and Andrew Tom to investigate how greenhouse tomato plants respond to water stress. California has been in a state of drought for several years now, which is a problem particularly because California is one of the largest agricultural producers of any state, using 80% of its water supply for agriculture. So the problem is that farmers need a way to minimize irrigation and conserve water while still maintaining optimum produce quality. The current most commonly used irrigation techniques are flood, drip, and spray irrigation, all of which entail the uniform watering of a field. However, soil and water conditions in a field tend to vary, so with these methods, parts of a field are being watered that don't need to be watered. For water conservation, the ideal method would be to water only those parts of the field that do need water. So the long-term goal of our research is the development of a sensor that manages irrigation for field crops based on their water needs. Doing this, however, entails being able to figure out which parts of the field it is that need water. So current methods for detecting water availability include measuring stem water potential and stomatal conductance of plants, as well as using soil moisture monitoring. Stem water potential is the tension on water within a plant, measured with a pressure chamber, and stomatal conductance is the rate of transpiration and gas exchange of a plant, measured with a barometer. And it's been established that when a plant is stressed, both of these values will decrease. So theoretically, if a farmer wanted to know if their plants were stressed, they could track these values over time and see if they decreased. However, in the context of large-scale agriculture, this isn't practical because to get an accurate representation of the whole field, they would need to take a measurement from every single plant in the field, which is incredibly time-consuming. A more practical technique would be to use soil moisture monitoring, where a sensor is inserted into the soil to measure water content, but this has been shown to be inaccurate as the sensor positioning influences the reading and often is not representative of the entire water availability. So what we do know is that when a plant's stressed, as we mentioned, stomatal conductance and stem water potential decrease. We also know that a plant will transpire less. And transpiration does a lot of things for a plant, but it also serves as a cooling mechanism. So if a plant's transpiring less, its leaves are going to be warmer. And leaf temperature is a more convenient thing to measure. Previously, a team of researchers here at UC Davis developed a sensor that measured leaf temperature for grape, walnut, and almond crops, which attach to the leaves of the plant. However, for herbaceous plants like tomato plants, this isn't practical because you can't attach the sensor to the plant. So what we looked at in our experiment was using overhead infrared canopy temperature sensors to monitor canopy temperature over time. Some things we were trying to find out were, firstly, which measurement is the most sensitive, so how do trends in canopy temperature compare with trends measured with conventional techniques? How fast does the response happen? For example, will stomatal conductance respond more quickly than canopy temperature? How does stress impact fruiting? Because in the context of agriculture, what really matters is whether or not the crop produces a commercially viable product. And finally, recovery time. So after the plant is done being stressed, how long will it take it to recover? So for our experiment, we used two similar tomato plants labeled IR1 and IR2, and we had scales that were continuously tracking the weight of the plant so that we could see how much water had transpired. We also had overhead infrared sensors that measured canopy temperature continuously. So every morning at 8 in the morning, for the first few days of the experiment, we watered both plants at flood capacity and measured the weight of each plant before and after watering. Then in the afternoons at 1 p.m., we would use a barometer to measure the stomatal conductance and leaf temperature of 3 leaves from each plant, and we would use a pressure chamber to measure the stem water potential of 1 leaf from each plant. After a few days of observing the trends in these variables, we would make sure that the plants were reacting in a similar way and then implement the stress conditions upon IR1. So to do this, we watered IR2 at flood capacity and then gave IR1 half of the water that IR2 was given. After about 3 days of stress, we would then begin to water both plants at flood capacity again and observe the trends. So these are the results we had for trial 1. This graph represents the stomatal conductances of both IR1 and IR2. The first vertical red line represents when the stress began, and the next vertical line represents when stress ended. The black trend line is the conductances for IR1, the stress plant, and the blue trend line is the conductances for IR2, the control. So we can see that before stress was implemented, the conductances for IR1 were higher than those of IR2, but once stress started, the conductances decreased significantly, and once recovery started again, they began to rise back up. We saw similar trends with stem water potential, so
before stress was implemented, IR1 had a higher potential than IR2, and once stress began, it dropped below that of IR2. And interestingly, when recovery started, about 4 days in, it went higher than IR2 again. So this shows that not only does stem water potential react quickly to the water stress, but it also recovers quickly. We also looked at the daily evapotranspiration of a plant, and IR1 was initially transpiring less than IR2, but once the stress began, it began transpiring even less. You can see the difference between them increases, and once recovery starts, this difference begins to decrease again. We also looked at leaf temperature, which were somewhat variable before stress was implemented, but it's clear that once the stress begins, IR1's temperature gets higher than that of IR2. So this graph is the leaf temperature minus the ambient temperature measured with the perometer on 3 different leaves. So the perometer attaches directly to the leaf. So for values that are above 0 on this graph, that means that the leaf temperature is warmer than the ambient temperature. And again, once recovery started, the difference between them began to decrease again. So for the first 4 variables, everything was going as planned. Everything reacted quickly to the water stress and began to recover. But what we were really interested in was, again, the canopy temperature measured from overhead, and this is what happened. So what we expected to happen was that IR1's temperature relative to IR2 would increase. So in that middle section, the black line would get a lot closer to the blue line and maybe even cross it. And this sort of happened. The difference between them decreased a little bit, but it's not easily detectable, and if you're a farmer trying to see if your plants need to be watered, this isn't exactly helpful. So the reason we hypothesized for why this happened is because of sensor positioning. So when we started the experiment, we had our 2 plants and the sensors overhead, but as the experiment progressed, the plants got taller than the sensors. So by the time we were into the experiment, the sensors didn't have a comprehensive view of the upper canopy. So in our second trial, we used younger and shorter plants, partially because of the sensor positioning, but also because younger plants transpire less and we wanted to see if it would have a slower response to the stress. We also extended the time period before stress was implemented to ensure that the plants weren't being stressed beforehand and there was no biasing effect. And we also counted the number of open flowers to make our experiment more relevant in the context of agriculture, because flowering can predict fruiting. And the trends we observed in stomatal conductance, stem water potential, leaf temperature, and evapotranspiration were very similar to those in trial 1, but this time with canopy temperature, it was able to predict the water stress. So you can see that before stress was implemented, that longer period, they're following pretty much the same trends, with IR1 having slightly lower canopy temperature than IR2. And then once the stress is implemented, IR1's temperature gets higher than that of IR2. And in recovery, it stays higher, but we had a very short recovery period this time, so this doesn't really tell us much about how the plant recovers. We also looked at the number of open flowers, which was very interesting because IR1 was initially flowering more than IR2. IR1's the stress plant again. And even as the stress was implemented, it continued to flower even at a faster rate. It did lose some flowers during recovery, but so did IR2. And this shows us that the water stress may not significantly negatively impact the capacity of the plant to develop fruit later on. So this experiment shows us that not only do stem water potential, stomatal conductance, evapotranspiration, and leaf temperature predict water stress in greenhouse tomato plants, but so can canopy temperature. And this indicates that later on, it might be possible to use a sensor to monitor canopy temperature to detect water stress in the field. Additionally, the fast recovery time and the ability of the plant to flower even after stress is implemented indicates that water stress may not have as big of an impact on the plant as believed. It's important to note, however, that this experiment took place in a fairly controlled greenhouse with 2 plants and 2 trials, so we can't really extrapolate this data to the field, which is why further research needs to be done. Currently, the engineering department here at UC Davis is working on refining a sensor that will apply in the field, so field tests with this new sensor would need to be done. Additionally, more trials with a longer recovery time would be good to observe how the different variables react in the long run and how the tomato produces fruit. Ultimately, we hope that our findings and future findings will contribute to a larger goal of water conservation. So I'd like to thank Dr. Ken Shackle for being my professor and teaching me about plants, Andrew Tom for being my mentor, and waking up at 6 yesterday to read my paper. Corinne for her counseling and having good vegan food at all our meetings. And Rick and Megan for making all of this possible. And everyone in the Young Scholars Program, especially Sithara for turning off the air conditioning last night. These are my references. Are there any questions?
So you get into the drone business.
Yeah.
I noticed according to graphs, it went from like the end of June to around July 9th. Is there a reason why you didn't collect your data until the end of the program?
Because we wanted to start a second trial, and we had already finished the stress conditions, and we did collect data till the end of this program in the second trial. Like July 26th. Any other questions? No? Thank you.
Lisa, we're about 5 minutes ahead. Is everybody here who's going to be here for you?
Yeah, but my parents are watching live stream.
Okay, so we'll wait.
Okay.
Okay, thank you.
So is your lab, are your lab people here?
Yeah.
Shall I see if your parents are ready?
You want to email them?
No, I'll chat with them.
Alright.
Oh my goodness. What are their names? Big Brother talking. It's Big Brother talking now. So, let's see. What's your mom or dad's name? Y-I-F-E-N-G. Okay. Are you watching?
Go back and 4 times or just
So let's see. See what happens. See how awake. Well, there's a chat window in YouTube. I am here. So are they both going to watch or just one?
Just my mom. Yeah.
That's you thing?
Yeah.
Then she's ready to go.
Hi everybody, my name is Lisa and I'm working under the mentorship of Dr. Kenneth Shackle in the Plant Sciences Department. And my research is focused on different methods used to test for water potential. In other words, water stress in plants. Stem water potential is basically a way of quantifying the suction that a plant places on water in the soil. And this can be expressed in units of negative pressure. According to the plant atmosphere continuum model, there's a tension within each plant that allows water to be transported from the soil of the plant into the stem of a plant, into the leaves, and into the atmosphere. And if you look on the diagram to your right, you can see that as you move up a plant, the water potential becomes more negative. And this gradient causes water to be transported throughout the plant. In the past, researchers have found a correlation between a plant's need for irrigation and its water potential. So typically in a plant that needs irrigation, the water potential would be a lot negative and compared to a plant that has been irrigated already. Stem water potential has a lot of uses in agriculture because it basically takes into consideration all factors that affect a plant's need for irrigation, such as the humidity of the environment, the last time it rained, the last time a plant received water, and puts this into one single number that allows growers to know exactly when they need to water their plants. As of now, however, there is no universally accepted method of measuring stem water potential, and because of this, a lot of growers use different variances and methods. The problem with this is that some variances might cause inaccurate measurements of stem water potential, therefore, which might affect plant health. If, for example, if a farmer decides to measure the plant and finds that the water potential is really high, they might think that the plant is unstressed, while in reality it might actually need water. So what I'm doing is that I'll be testing the different variances and methods that these growers use to figure out whether or not they have any impact on accurate stem water potential measurement. The materials that I used in my research were two types of pressure chambers, and both of these chambers use compressed nitrogen gas that I could add into the chamber to look at stem water potential. And also worked with 4 plant species, the cherry, the grape, pistachios, and walnuts. And also used light-proof aluminum bags and magnifying lens. The basic procedure in my experiment is that first I would bag 2 to 3 adjacent leaves on a tree, and then after bagging them, I would wait around 10 to 15 minutes before I cut the leaves off of the trees using a razor blade, and then putting it into a pressure chamber with the petiole sticking out. So if you look at step number 3, you can see that there's a little petiole sticking out that I can view that would help me measure stem water potential. And then after this, I would turn on the pressure in the pressure chamber and then turn it off when I notice that water visibly leaves the xylem of the petiole. And the pressure in the chamber at which this happens is the absolute value of the stem water potential of the plant. The different variations and methods that I tested with my experiment is cutting versus recutting. So some farmers would cut a leaf directly off of a tree and then place it into the chamber, while others might pull a leaf off and then cut it once and then cut it once again so that it could fit better inside of a chamber. Another method that I'm testing is the position at which the leaf is bagged. So there might be a difference between bagging a leaf that is closer to the stem of a tree versus a leaf that is far away from the stem of a tree. And then lastly, I did a test for folding versus non-folding. So basically with a lot of plant species like grapes, they tend to have leaves that are too large to fit inside of a pressure chamber. So I'm testing whether or not folding a leaf into a pressure chamber has any impact on the measured stem water potential. And with these different methods, the reason why I bag adjacent leaves is that adjacent leaves tend to have the same water potential. So if I do one treatment on one leaf and another treatment on another leaf, then I can see whether or not there is a difference in the measured stem water potential. So the second half of my research is focused on pistachios. So with pistachios, there's a problem that a gooey substance leaks out when it is cut, and this can often hinder accurate stem water potential measurements. So with this, I'm doing 3 different treatments on pistachios to figure out whether there's a clear method of testing for stem water potential. So in the black circle, I circled a branch with the leaflets off of it. So what I would do is that I took a branch off, stripped it of its bark, and then put it into a pressure chamber. And then the second method that I tested was bagging just a terminal leaflet, which you can see in the red circle. And then the third one is just taking a branch off of a tree without bagging it at all, because I wanted to see the amount of difference it has on the stem water potential measured. So every day I would do 8 to 10 trials, and each trial compares 2 to 3 methods, so whether the leaf is unfolded or folded. And then after that, I would put it into a statistical analysis system that would return the mean value between each different method that I tested and the p-value. So first, I'll be talking about my results for near versus far. So the column that I'm focusing most on is a column that says PR is greater than F, and this gives you the p-value of my experiment. So if a p-value is less than 0.05, it means that there is a significant difference in the measured stem water potential between 2 different methods. So here we can see that the p-value, which I highlighted, is 0.0003, and this is significantly lesser than 0.05. And because of this, we can conclude that leaves that were bagged near to the stem of a tree had a significantly different stem water potential than leaves that were bagged far away from the stem of a tree. And then if you look at the table underneath that, you can see that there is, I put the means of each treatment that I did, and you can see that there's a significant difference between the means of a leaf that is bagged near to the stem of a tree versus a leaf that is far away. And then next, I did the same test on cherries, and once again, it returned a very low p-value, meaning that there was a large difference between leaves that were near to the stem and leaves that are far away. And then for second, I did a recutting test, which is like cutting a leaf directly off of a tree and then putting it into the chamber, versus cutting a leaf off and then cutting it once more and then putting it into the chamber. So here I found that the p-value was 0.74, which is a very high p-value, which means that there was no significant difference found between cutting a leaf versus recutting a leaf. And then if you look at the mean values, there's less than a 0.1 bar difference between just a cut leaf versus a recut leaf. And then here is a graph of the cherry tree that I tested over the course of a day. So the x-axis shows the different time periods that I did the testing on, and the y-axis shows the stem water potential. So as you can see in this graph, sometimes the cut leaf would have a higher stem water potential than the recut leaf, while other times the recut would have a higher than the cut. So this shows no overall trend in whether or not recutting causes any high stem water potential. So it shows that generally recutting and cutting has no significant impact on cherry trees. And then next I'll be talking about folding. So I did my first folding test on cherries, and these were 2 tests done on 2 separate days. And I found that the p-values were all very high for these tests, so 0.6 and 0.3. And this shows that there was no difference between a folded leaf and an unfolded leaf. And with grapes is a really large concern because grape leaves tend to be very large and difficult to fit into a pressure chamber. So it would definitely be more convenient if growers were able to fold a leaf into the pressure chamber instead of buying a larger pressure chamber that they could put an unfolded leaf in. So with grapes, I did 5 tests in total. And for the first test that I did, I found that the p-value was 0.6. And this is, once again, a very high p-value, showing that folded versus non-folded had no difference in stem water potential. However, on the second test that I did, I found that the p-value was 0.003, which means that in this test specifically, there seemed to be a difference between an unfolded grape leaf versus a folded grape leaf. So overall, as I said earlier, I did 5 tests in total, and 2 of these tests found a significant difference between an unfolded leaf and a folded leaf, while the other 3 found that there was no difference. And out of these 5 tests, 3 tests found that a folded leaf had a higher stem water potential than an unfolded, while the opposite was true for the other 2. So once again, I noticed no overall trend between folding versus unfolding. So therefore, I concluded that folding generally didn't have a large impact on grape stem water potential. And with pistachios, as I said earlier, I tested a bagged petiole versus a bagged branch versus an unbagged branch. And here we can see that the p-values are very tiny, showing that the 3 different methods tend out to have very different stem water potentials. And if you look at the tables, you can see that the bagged petiole and the bagged branch tended to have very close stem water potential measurements, while if you look at the unbagged branch, it would typically have a water potential of a 10 bar difference between the first 2 bagged. So I realized that unbagged tended to be more inaccurate and gave lower stem water potential measurements than a bagged petiole. And additionally, the problem with a bagged petiole that I was testing was that oftentimes the gooey substance would leak out, and out of the 8 trials I did in a day, 4 of them would be very uncertain. And with the bagged branch, however, the endpoints were very clear and the measurements for stem water potential were very precise. So in conclusion, I found that folding and recutting generally had no impact on stem water potential. And this is good because for farmers, they don't have to worry about whether or not they have to leave the leaf unfolded in the pressure chamber or whether or not they have to just cut a leaf directly off and make it right the first time. So with this, they can customize these different methods to their own liking, to their own convenience. And additionally, they won't have to worry about buying a larger pressure chamber, which is very costly. And then second, I found that far and near leaves did have a difference in stem water potential measured. So with the data I collected, I would suggest that growers standardize where they bag every leaf on the trees so that it would be around the same for each plant that they use. And then with pistachios, there was generally no difference in the stem water potential between a bagged petiole versus a bagged branch. But with the bagged branch, however, the stem water potential measurements were a lot clearer and a lot easier to distinguish, so that's why I would suggest using a branch instead of a petiole to test for pistachios. And then also, an unbagged leaf tended to have a very low stem water potential, and this may be inaccurate, and therefore it is unadvised to use an unbagged leaf at all. So as of now, my results are conclusive for only 4 plant species. So future research should be focused on figuring out whether or not folding and recutting had any differences for other plant species as a whole. And additionally, further research should be dedicated to finding the most accurate stem water potential measurement so that all farmers know exactly when they need to water their plants and exactly when their plants are stressed. I would like to acknowledge Dr. Kenneth Shackle for his constant supervision and guidance and helping me with my project. And I would also like to thank Corinne Rushing, Jiong Fei, Andrew Tom, and Michael Rawls for assisting me with my project. And lastly, I would like to express my gratitude to the entire Young Scholars Program and all of the staff and participants for giving me such a wonderful experience here. And then here are my works cited and the photos that I used. Thank you. Any questions, Alex?
Were
Why did you pick those plants specifically?
Okay, so for cherries, they had the xylem was very visible and it was very easy to tell when the water potential has been reached. So that's why I did cherries, because it would be easy to do folding and recutting tests on them. And then while for grapes have very large leaves, so they would be a good measure as to whether or not folding had an impact on different plant species. And then Nathan?
Can you go back to your results slide?
Yeah. Which one?
Discussion conclusion, never mind. You talked about far and near. Could you go over that one more time?
Yeah, so generally leaves that were far away from the stem of a tree, they tended to give lower stem water potential measurements than leaves that were near to the stem of the tree. And as I said earlier, there's no universally accepted method of measuring stem water potential, so we don't know the exact stem water potential of the plant. But however, I would suggest that growers standardize where they measure the stem water potential in each tree, so that way they can get the same measurements every single time that they do it. David?
You said when pistachio leaves are cut, they excrete a fluid. Is that what latex is?
It's so as of now there isn't a lot of research about latex and what pistachios leak out when they cut, but generally it's something that's very similar to resin. So yeah. Ben?
So can you go back to your folding with the grape leaves? Or folding grape, this. So you had two tests that found a significant difference. And then you have two different types that, you have two different results. You have the folded is greater than unfolded and unfolded is greater than folded.
Yeah.
Did the two tests who did find significant differences show the same result or a different result?
Oh, you mean in the greater or less
than? So with your two tests that gave a difference, or that said there was a difference, were they the same difference or were they different?
No, it wasn't.
So it was one of them said folded was greater than unfolded. Yeah. Okay, that's interesting.
It is. Any other questions? All right, thank you guys.
So we're about 3 minutes early. Are the people who are coming to see you here? Okay, we'll give you a couple minutes. That's fine.
The way in which we Sarah's at school. Yeah.
I know we're missing one.
I never got it back. I told her I'd get her a break next week. So we talked to the pistachio place.
I think we're missing somebody else.
And they are lenticifer. I'm not sure.
It helps if I stretch it.
Oh yeah, I found something that said it was
Yeah. Yeah, I guess Anthony.
No, I don't think so.
She said, put take
Intentional?
Not even nice to. I just had two. I made it
Maybe. We're only missing.
I've been.
I just hold my head like this.
It's like one of those those brushes for makeup.
Oh
I got butterflies in the yeah, they're 41. I got butterflies in the face and they get where you delete. Butterflies, I know. Well, it's because these are almost done. You know, ran out of fingers and toes. Come on. Yeah, I figured it out. Yeah, smart. I figured it out. Yeah, I know.
Oh my god.
I do that too. Like your kids.
I'm gonna take a nap.
Finish it. Done. Period. Yes, sir. Over practice. Well, some of them don't quite feel you have butterflies? When you put them like I'm a little bit used to presenting them not. No, just so all the butterflies starting to one face and the overall I'll be really nice. You did a great presentation.
We may be late, but we try never to be early. Because there are people, we don't know who's watching.
Hi, my name is George, and in the past 6 weeks, I've been working in Dr. Brown's lab on the Department of Plant Sciences and under supervision of Mazia Campos and Francisco Acevedo. So, the purpose of this research is to investigate the influence of salinity on water status of almond. So, salinity is known to compromise the plant-water relations in many species, and almond is a particularly sensitive plant. However, not enough studies were done to be able to quantify its salinity tolerance. So, and the recent drought issues in California also made this issue to be solved more urgently, because as less snow water is available, the agricultural industry has to utilize the underground water, which has a much higher salt concentration than snow water. So, as I mentioned before, I'm trying to solve the question, what is the effect of salinity on water status in almond? Water status is basically the stem water potential, and the sum of pressure potential and osmotic potential is equal to water potential. So if salts are applied to the soil of the plant, the water potential of the soil would decrease, which would make the almond trees to try to internally adjust its osmotic potential, therefore the water potential throughout its system. We hypothesized that if salts were applied to the soil, it would force the almond tree to adjust internally and therefore have a lower stem water potential. This is because when the plants are trying to adjust its water potential throughout itself, it's going to utilize the carbohydrates or the sugars within its body, and to balance out the osmotic potential, which is correlated with the water potential. However, when this process occurs, they're using its energy. Therefore, their growth rate may decrease. So this is why the growth rate of almond trees so decreased. So some previous research has also suggested that there may be those 3 factors may affect the growth rate of almond trees. So the first one is ion toxicity, which is the accumulation of salts in the leaves. The second one is ionic imbalance, in which the sodium ions competes with the potassium ion, which is a source of nutrient, and the chloride ions will degrade the chlorophyll, which is essential to the conversion of solar energy to usable energy. The third one would be osmotic stress, which will be which is also the salinity stress. So, like I mentioned before, when the almond trees are trying to adjust osmotically in order to balance out its water potential throughout the system, it is also losing its energy, therefore have a lower growth rate for growth. So I conducted this research at the plant pathology field house on the UC Davis campus during the period of July 15th to July 26th, and there was no rainfall during this period. On the left, it's the experimental almond tree that I used, and on the right is the control almond tree that I used. So each tree was irrigated with 8 liters of water and then in the experimental tree, they were irrigated with water containing 40 millimoles of salts, while in the control group, no salts were applied. All the measurements of stem water potential were taken in the at the midday, which is around 12 o'clock, and 5 leaves were selected from each tree. The selection of leaves was based on 3 factors. The first one is the petiole length, the second one is the level of browning on the leaves, and the third one would be the distance from the stem to the leaves. And after the selection of leaves, all 10 leaves were bagged with this aluminum foil bag, and left on the tree for at least 15 minutes until collection. So before I go into the later procedures, I would like to introduce you the Scolander pressure chamber, which is used to measure the stem water potential. So in here, it's the cap of the pressure chamber where the leaf will be inserted in. Here, this is the on and off switch. This is the knob that controls the rate of pressure application, and here is where the stem water potential would show. So after the collection of leaves, I cut the tip of the petioles to expose the xylem, and then I'll insert the leaf into the cap of the pressure chamber like this, and put it back to the place. Then I'll turn on the switch and use a magnifying lens to observe the xylem of the leaf. While observing, I'll use this knob to adjust the rate of pressure application as desired. When the first water droplet comes out of the xylem of the petiole, I'll immediately turn off the Scolander pressure chamber and then record the stem water potential value. After that, I'll exhaust the pressure and release all the pressure inside the pressure chamber and do the next sample. So all the data were all the stem water potential values of 5 leaves were averaged and plotted on this graph. As you can see, during the first 10 days, there was no significant increase or decrease. However, approaching the end, there was a decrease in the stem water potential values. I then plotted the stem water potential value on a normalized graph. This was normalized by dividing the experimental or control group by only the control stem water potential. Therefore, the control group should always have a constant of 1. As you can see from the graph, the experimental has had a increasing normalized stem water potential ratio. Therefore, this means that the difference between the experimental stem water potential and the control water potential is increasing over time. So, in my hypothesis, I stated that the application of salt would decrease the stem water potential values. It was shown only in the after 10 days of salt application, but not the first 10 days, as there wasn't that much fluctuation in the first 10 days. This may be because the almond tree needed time to adjust to start osmotic adjustment. However, if there are more evidence that will be able to back up the last decreasing trend, it will be better. Therefore, a long-term research will be much better than short-term research like this. So some of the application of this research will be to quantify the salinity tolerance of almond trees. It is important because the agricultural industry wants to optimize the amount of salt that's needed in order to optimize the growth and yield while not damaging the almond trees. This research has also a significance to the science world. This provided a data set for this particular salt concentration on a salt concentration versus stem water potential graph for almond trees. Some future research would be to vary the salt concentration and discover its effect on growth and yield, therefore creating a growth and yield curve. Ion toxicity would also be very important to be investigated, as it is a crucial factor that may decrease the growth rate of almond trees. Lastly, I'd like to acknowledge the following people for helping me throughout the process. Thank you to Dr. Patrick Brown for allowing me to be at his lab, and thank you to Dr. Mazia Kentlis for helping me throughout the process, and thank you to Francisco Acevedo for give me advice and let me in his greenhouse. And I would like to acknowledge all other lab members as well. I'd also like to say thank you to Dr. Rick Pomeroy and Megan McKenzie, Stephen Ramsey, and all other counselors, as well as all of the YSP students. Thank you. What questions do you have?
What was your reasoning for analyzing like the growth of leaves at midday?
Because that's when the stem water potential is the most stabilized and at its maximum value. Ben?
How did you pick a 40 millimolar concentration? Or
It actually doesn't matter because I don't have any variation in this situation, so it can be 30 or 50. I just needed to investigate the effect of salinity on almond tree.
Okay.
But variation would, in the future, there may be some variation research that will see how much concentration salt that may affect almond tree. Renee?
So do you know why stem water potential is most stable at noon?
I believe it's because of its temperature, but I need to do more research on the question.
Maybe I missed this, but on the first day that you made the measurement, then there was no salinity treatment yet?
There was, actually.
Could you go back to the slide where you showed just the stem water potential, back even before, yeah, there. So when was the salt actually applied?
On the 13th. On the 13th of July.
Okay. Oh, okay, just a couple days before.
Yes.
So, but already by the 15th, it looks like there's a little bit of a difference.
Yes.
But it wasn't significant?
It wasn't large enough to be able to conclude. Thank you. Next, I would like to introduce Alexandra Lee.
Alexandra, we're about three minutes ahead of students who are coming to see you here. Okay.
Yes. Let me check.
Why?
Yes.
Great.
Now I see it.
Good morning, everyone. My name is Alexandra Lee, and for the past 6 weeks, I've been conducting research regarding the synthesis of 2,3,4,5,6 pentafluorobenzene diazonium tetrafluoroborate and its possible function as a p-type dopant. And from now on, I'll be referring to this compound as PFBDT. I've been working under the guidance of Dr. Mark Mescal in the Department of Chemistry and directly under graduate student Joseph Beckett. Now, before I move on to the lab goals of producing this diazonium salt, I'll go over some of the common properties of it. Diazonium salt is a unique salt that contains a common functional group, an N triple bond that contains the positive charge. Also, diazonium salts are usually aromatic, which means it has a conjugated system with the pi bonds. On the figure on the left, you can see a common cation of the diazonium salt, which is called the benzene diazonium cation. As you can see, it has the N triple bond with the positive charge, and it is an aromatic compound. The figure on the right is the diazonium salt, PFBDT, and as also you can see, it is an aromatic compound with the N triple bond with the positive charge. And the reason why we are trying to synthesize PFBDT is to see if it can act as a dopant. Now, dopant is a substance that allows an agent to conduct electricity, and there are two types. An N-type dopant is one that adds an electron to conduct electricity, and a P-type dopant is one that removes an electron. This creates a hole that can move in a sea of electrons, which also acts as an electric current. Now, most people think of doping in semiconductors as doping in inorganic semiconductors, as you can see in the image on the left, in silicon, but we'll be testing whether PFBDT will be able to act as a P-type dopant in organic semiconductors, specifically P3HT. Now, we believe that PFBDT will be able to extract an electron because of its highly electrophilic nature. If you look back to the structure of PFBDT, it has many fluorine groups, and these are very electronegative, which means it has electron withdrawing symptoms. If it can withdraw an electron from the electron-rich organic polymer, P3HT, then it will be able to act as a P-type dopant. Now, in the past, organic compounds have been shown to have doping effects on organic polymers. For example, F4TCNQ in figure A and 4-BBT in figure B. Now, there are some limitations to these P-type dopants. For example, F4TCNQ, although it is known as the standard dopant, it is highly insoluble in organic solvents. Now, it is important that these dopants are soluble in organic solvents because it may be able to dissolve and then become the spray coating for the film, which is used to dope the organic polymer. And as for 4-BBT, research has shown that it does have doping effects on mechanically exfoliated graphene, which is an electron-rich organic polymer, but it is very limited. Now, we believe PFBDT in figure C will be able to be more efficient than 4-BBT because of its highly electrophilic nature. It has more fluorine groups in its cation, which means it has a greater efficiency to withdraw that one electron to create the electron current. And it is less nonpolar than F4TCNQ, so it is expected to be more soluble in these organic solvents. Now, our goals in the lab are to find a more accessible route to synthesize PFBT, and once it has been purified and its identity has been confirmed, we will be studying its stability and its decomposition chemistry. Now, after it has been synthesized, chemical engineers will be able to study if it can act as a P-type dopant and its practical applications. Now, we're a synthetic chemistry lab, so we won't be directly studying its applications as a P-type dopant, but this is the larger application of synthesizing PFBDT. There have been recent interest in organic semiconductors over inorganic semiconductors because they're rather different. As you can see in the image above, inorganic semiconductors are held together through covalent bonds, and thus they're rigid semiconductors. Whereas organic semiconductors here are held together by weak van der Waals forces, and this allows them to become flexible and thinner. As you can see in the photo on the left, this allows for the electrons to travel in a non-linear fashion, and therefore it will be able to be flexible. Also, organic semiconductors have easier processing, and this allows for the cost of organoelectronics to be more cheap than inorganic ones. There are many applications for organic semiconductors. For example, OLEDs are organic light emitting diodes. And these are the two photos on the right, and you can see there are many applications in the technology in very thin screens and flexible screens that we can apply in the future. And with a more efficient P-type dopant, this will allow for the OLEDs to become more efficient as well. Also, solar cells are cheaper with the application of organic semiconductors, and this will allow for a more renewable future if allowed to be applied. So in synthesizing PFBDT, Schlenk techniques are used in which the reactions are carried out in either argon or vacuum. This, because of its highly electrophilic nature, if it is exposed to air or vacuum, it will decompose. So you can see in the photo, the manifold in the center allows for the reactions to be carried out in either a non-reactive gas or vacuum. Also, after it has been synthesized, we will analyze the compound through NMR, nuclear magnetic resonance, or IR, infrared spectroscopy. So this is the reaction we did. We started with pentafluoroaniline, added the hydrofluoric acid and ether complex. As you can see, that is what provides the BF4 for the anion of the compound, and added isoamyl nitrite. After this reaction has been carried out at negative 41 degrees Celsius, this allows for a white precipitate form, which is PFBDT. And when it is synthesized, we do wash it with diethyl ether and put it under high vacuum, but this doesn't purify the compound completely. So purification is taking place. the method we use is called vapor diffusion. And in vapor diffusion, the product is placed in a solvent that it is soluble in and is surrounded by a solvent that is insoluble in. Now, over time, the vapors from the solvent that is insoluble in will travel into the original solvent, and this will change the polarity of the solvent that the product is in. As it becomes insoluble in this new solvent, the crystals will form slowly, and that is what helps form the crystals. So as a result, some yellowish crystals formed after recrystallization, and we confirmed its identity through a proton NMR. And ideally, the proton NMR will not have any peaks, as the compound does not have any hydrogen in it. But the one major peak seen is at 1.94 ppn, which represents the solvent. And this is not to be worried about because we know its identity is the solvent and not the product itself. The other traces are of the starting material and the solvent, but they're very little and to be taken account for. So this is confirming the identity of the compound. We also took a fluorine NMR, and these have 4 peaks which all correspond to the fluorines in the compound. So although zoomed out, each peak looks like one single peak, each peak actually has splitting with sub-peaks. And to identify how many peaks that one peak has, it is the number n+1, in which n is the number of fluorines adjacent to the fluorine that corresponds to that peak. So for example, for the first one, it has the integration value of 1, and the integration value corresponds to the ratio of fluorines per group to the other fluorines. As you can see, there are 5 subpeaks, which means that there are 4 other fluorines that are adjacent to the fluorine. So this only corresponds to this one fluorine. Next has the integration value of 2, and this corresponds to these 2 fluorines. And the next peak has the integration value of 4, and this corresponds to the fluorines in the anion. Lastly, the peak with the integration value of around 2 corresponds to these 2 fluorines. We also took an IR infrared spectroscopy to confirm the identity of it, and most particularly to note is the N triple bond peak, as each peak represents a certain bond in the compound, so that occurs at around 2,800. So we have confirmed the identity of the compound, and we have also confirmed it through X-ray analysis of its crystal structure. Next, the solubility of the compound was analyzed. Most notable are that the compound is insoluble in DCM and soluble in acetonitrile and DMSO. For example, for the vapor diffusion, the sample was put in acetonitrile, and the anti-solvent was dry diethyl ether. Next, we studied the stability of PFBDT. As a powder, it was placed on argon and left in the counter, and after a few days, this white powder was turned into sort of a yellowish red powder, which is visible decomposition. With this, we deduced that with the addition of heat, as a rise to room temperature, the compound decomposes. Next, we studied the decomposition chemistry of the crystal itself. In the air, after left in the air in 24 hours, it turned into this red foamy substance, and when put into the NMR, it did not really show any results. So with this, we deduced that the compound polymerized and therefore could not be seen by the NMR. And when mixed with water, it creates this yellowish, viscous substance. And with this, we ran a couple NMRs, and there are 2 peaks in the fluorine scan, which indicated that it reacted with the water at the fluorine across from the N2 group. Lastly, we looked at its decomposition chemistry when exposed to heat. So its melting point is around 124 degrees Celsius to 131 degrees Celsius with a ramp of 4 degrees Celsius per minute. And also, it just, as you see the temperature increase in the melting point apparatus, you can visibly see the color getting darker, which is signs of decomposition. In summary, the new procedure created to synthesize PFBDT has confirmed its identity through IR and NMR. It is stable under argon and when kept at lower temperatures, and it is moderately soluble in organic solvents, though much more soluble than FR, TC, and Q. In the future, we can do research about why the powder turned yellow when recrystallized, and more broadly, other stable dopants that can be more efficient in its applications to organoelectronics. I'd like to thank Dr. Mark Mascal to letting me work in his lab this summer. Also, I'd like to thank my graduate student for dealing with all my questions and messing up all the time. I'd like to thank my counselor Corinne and the directors of YSP, Megan and Dr. Rick Pomeroy. I'd also like to thank everyone here for helping me have such a great summer experience, and my parents for letting me live across the country for 6 weeks and for being here today. Thank you.
So you said that these organic semiconductors were useful in like solar panels. I remember the efficiency, information might be dated, but for organic solar panels is relatively low. Has that changed with it?
No, so the disadvantages of having organic semiconductors is that its efficiency is much lower than organic ones, and that's one of the reasons why people don't use organic ones. But as we can increase the efficiency through the type of dopants, that will also increase the efficiency of the organic electronics, if that makes sense.
Okay, yeah.
Brian?
For the spectroscopy of the spectroscopy side, like that you like circle the fluorines.
Oh yeah.
How did you determine which fluorines, which peaks were certain fluorines?
Okay, so I did this kind of quickly. So on the y-axis of the NMR, the more left you are, the more upfield you are, is kind of this lower the electron density of the particular element. So these are in order, 1, 2, 3, 4. The first one has the lowest electron density, and you know that this is the fluorine across from the N2 group because it has the lowest electron density, and also with the splitting, as you can see, there's like 5 peaks, and this is the yellow one is the only fluorine with 4 of them around it. And also, like, you can use the integration values to help you figure out which of the fluorines are in the same group as it corresponds to the ratio of the fluorines in the compound. Lily?
Is there a reason why the first and third graphs are like symmetrical, but the second one isn't?
You mean the third graph isn't?
Oh, I mean the first and the top and bottom one are like kind of symmetrical, whereas the second one are not for the.
Oh right, so for the blue one, the one that integrates to a value of 4, usually peaks should be symmetrical, but we believe, as indicated by the blue dotted line, that there's some like dipole interactions as the fluorine on the tetrafluoroborate has a negative dipole, whereas the N has the positive charge, so that one fluorine is a little more attracted to the N2 group, which causes the splitting of the peaks, because the ratio of those 2 subpeaks in the one with the integration value of 4 is around 1 to 3. Ben?
So you briefly explained what a dopant was. Can you go over what it is again? So it's the structure that adds and subtracts electrons. So this was right at the beginning. Yeah.
So as you can see the first one, there's like an extra electron next to the red dot,
Sure.
and then in the second one there's a hole, and like most people think of like conducting electricity as one electron moving around, but electricity can still be conducted if there's a hole moving around in a sea of electrons, and that's a p-type dopant.
Okay, so so dopant is a semiconductor?
No, it is one that either adds or subtracts the electron. The conductor itself is the one with the hole or with the additional electron, if that makes sense.
Okay.
What was the product yield for the reaction?
So when we took the yield of the powder, it was about, I believe, 89%, but through crystallization, I also took the yields, and they varied greatly from 23 to 64. It was mainly because we didn't allow for all the crystals to crash out before calculating the yield, so that wasn't really accurate. But I know that the yields do go down after recrystallization, but it's worth it for the stability of the compound. Thank you.
Thank you, ladies and gents. We are on our lunch break until 1:30.